During prophase, the first phase of mitosis, the duplicated chromosome condenses and becomes visible. Centrioles move to the opposite sides of the nucleus and help organize the spindle. The spindle forms DNA strands attached to a point called the centromere. The nucleus disappears and the nuclear envelope breaks down. During metaphase, the second phase of mitosis, the centromeres of the duplicated chromosomes line up across the center of the cell. Spindle fibers connect the centromere of the chromosome to the two poles of the spindle. During anaphase, the third phase of mitosis, the centromeres of the pulled apart and the chromatids separate to become individual chromosomes. The chromosomes separate into two groups near the poles of the spindle. During telophase, the fourth and final phase of mitosis, the chromosomes spread out into a tangle of chromatin. A nuclear envelope reforms around each cluster of chromosomes. The spindle breaks apart and the nucleus becomes visible in each daughter cells. Prophase 1. Each duplicated condensed chromosome pairs with its homologue. Homologue swap segments called crossing over. Each chromosome becomes attached to microtubules of newly forming spindle. Chromosomes are pushed and pulled into the middle of the cell. Sister chromatids of one homologue orient toward one pole and those of other homologue towards opposite pole. The spindle is now fully formed. Anaphase 1. Homologous chromosomes segregate from each other. The sister chromatids of each chromosome remain attached. The chromosomes arrive at opposite poles. The cytoplasm divides in now two haplid cells. Prophase 2. Microtubules attached to the connectors of the duplicated chromosomes. Motor proteins drive the movement of the chromosomes towards the spindle's equator. Metaphase 2. All of the duplicated chromosomes are lined up at the spindle equator, midway between the poles. Anaphase 2. Sister chromatids separate to become independent chromosomes. Motor proteins interact with microtubules to move the separated chromosomes to opposite poles. The chromosomes arrive at opposite ends of the cells. A nuclear envelope forms around each set of chromosomes. The cytoplasm divides. There are now four haplid cells.